Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing with you the photosystems and the role of accessory pigments and Calvin cycle and how the carbon dioxide molecules and all produce the triphosphate and what's the ratio there. So let's start. What are photosystems? Photosystems are light harvesting clusters. They harness light energy and they deliver it to the reaction center. Photosystems are located on the thylakoid membrane inside the chloroplast, like on the surface of this thylakoid membrane. And what one photosystem is having, this is the structure of one photosystem from the thylakoid membrane. This is the reaction center. The reaction center will have one molecule of a primary pigment. The primary pigment will always be chlorophyll A and nothing else but chlorophyll A. Then there are hundreds of molecules of accessory pigments being present there on the same photosystems. And these accessory pigments, these include chlorophyll B, these include xanthophyll, chlorophyll B, xanthophyll, beta carotene. These are all accessory pigments. So these are all photopigments. So this is how a single photosystem look like. There are two photosystems there, photosystem 2 and PS2 and PS1. PS2 absorbs, PS2 is having a chlorophyll a specific chlorophyll A that absorbs the light of wavelength 680 nanometer. And PS1 is having another form of chlorophyll A as the primary pigment. They both have the primary pigment chlorophyll A, but the wavelength that is absorbed at the chlorophyll A is different. For PS2, it is 680 nanometer. The chlorophyll A absorbs 680 nanometer, and PS1, the chlorophyll A is an isomer of another of the same chlorophyll A. It absorbs the light of 700 nanometer. So this is the main reaction center. This is where the electrons will get excited, and once the electron gets excited, they trigger electron transport chain that sets in motion the non-cyclic photophosphorylation that I've already covered in other videos in the same playlist. So how the photosystems work, we know the accessory pigments, what kind of um, wavelengths do they absorb? The chlorophyll A, chlorophyll B, they absorb yellow, green, and blue, green respectively, whereas carotenoids absorb orange and xanthophyll absorbs yellow. The beta carotene absorbs orange and xanthophyll absorbs yellow. So what exactly is happening? Why do you need the accessory pigments to be there when the chlorophyll A is already there? So and the reaction center is the chlorophyll A. So what's the role of the accessory pigment? The accessory pigments, they absorb the light energy wavelengths which are not absorbed by chlorophyll A. Already discussed with you that this chlorophyll A absorbs either 680 nanometer or 700 nanometer. What about the other wavelengths? These wavelengths are absorbed by by your accessory pigments. They harness the light energy delivered to the reaction center in PS1 and PS2. The reaction center is chlorophyll A. And once the chlorophyll A receives this light energy, the electron in them gets excited. When the electron gets excited in chlorophyll A, it triggers the ETC in thylakoid membrane. ATP is produced, reduced NADP is produced by the process of non-cyclic photophosphorylation, which then later on is used in Calvin cycle. We know in Calvin cycle ATP energy is used and reduced NADP is used. We already have covered how this helps and this works. You can recall how the photosynthesis stages, the light dependent reaction goes on, and non-cyclic photophosphorylation, cyclic photophosphorylation can go on. How the PS2 and PS1, they have this light, they absorb the light energy, the electrons get excited, the electrons go to a higher energy, and this is the electron transport chain that occurs. This electron transport chain allow the electrons to move down the energy gradient, they lose energy, chemiosmosis occur, and ATP gets produced. And eventually the electrons are passed on from, in non-cyclic photophosphorylation, the electron of one are passed on to the other. For instance, how it goes about, the electrons in PS2, they go to a higher energy, and the replacement electrons come from photolysis of water. Photolysis of water occur because there is an enzyme present here in PS2. It splits water molecule. When it splits water molecules, the electrons of water splitting comes here in the PS2. At the same time, PS1 loses its electrons, they go to a higher energy, and these electrons fall into NADP, and the 2H plus from splitting of water, they get added to NADP as well. This reduces NADP in the stroma. PS1 receives the replacement electron that actually belong to PS2. So this is how the flow of electrons is non-cyclic. So in non-cyclic photophosphorylation, we have oxygen being produced as a waste product. We have AT being produced. We have reduced NAD being produced as well. When cyclic photophosphorylation occurs only in PS1. And when this occurs, The electrons from PS1, they go to a higher energy and they come back again. This occurs when reduced NADP is existing as reduced NADP and it is not oxidized. So if reduced NADP keep existing in the reduced form, it cannot accept the electrons coming from PS1 any further. So PS1 will have to allow its own electrons to come back to PS1. This ends up continuing cyclic photophosphorylation. When cyclic photophosphorylation occurs, electron transport chain continues, chemiosmosis occur, and ATP is produced. So in cyclic photophosphorylation, only ATP is produced. Reduced NADP is not further produced. Oxygen is not produced because photolysis of water is not occurring. So this is something that I was reviewing with you. So moving on to Calvin cycle. What is Calvin cycle? In Kelvin cycle, we already know that this occurs in the stroma, and carbon dioxide is fixed into sugars, and the sugar that is produced is called triose phosphate. The enzyme Rubisco is used in the stroma. There's already RUBP, which is a 5-carbon compound. It combined with carbon dioxide, which is a 1-carbon compound, to produce a 6-carbon unstable intermediate. This unstable intermediate quickly disintegrates into two molecules of GP. GP is glycerate 3-phosphate. So two molecules of glycerate 3-phosphate are produced. Now, these two molecules of glycerate 3-phosphate will be converted to TP, which is triose phosphate. Two molecules of TP will be produced, again having 3 carbon each. This reaction continues by consuming the products of light-dependent reaction. The reduced NADP, which was produced in non-cyclic photophosphorylation, and the ATP that was produced uses up here. So the reduced NADP gets oxidized. This reduced NADP, once oxidized, will go back again, and it will allow the same non-cyclic photophosphorylation to keep on getting continued. So eventually TP is made here. And once TP is produced, this TP is the 3-carbon compound. Using additional energy, this TP can be converted back into RUBP to regenerate RUBP. So this cycle can go on. And this TP can be converted to glucose, fructose, cellulose, sugars of pentoses, which is ribose, deoxyribose, fatty acid, acetyl. And yes, RUBP obviously is regenerated through the same Calvin cycle. When Calvin cycle is occurring, what actually is going on? This is the math of the Calvin cycle. So each Calvin cycle is fixing one carbon dioxide molecule we know to produce two molecules of TP. The two molecules of TP are being produced. So 6 carbon dioxide we know are used to synthesize one glucose molecule, because 6 carbon dioxide will end up having 6 carbons. We know the glucose molecule is having 6 carbon, CH6CH12O6. So Calvin cycle should occur 6 times for one glucose molecule to be produced. Now, when this is occurring, 6 cycles produce 12 TP molecules, since one cycle produces 2 TP, so 6 cycles produce 12 TP molecules. Two TP molecules can join to form one glucose molecule, because one TP is having 3 carbons. When you add 3 carbon with a 3 carbon, you get a 6-carbon compound, which is glucose. So 10 TP molecule will be left behind. 10 TP molecule will have 30 carbons, because one TP is having 3 carbons, so 10 TP will have 30 carbons. These are used to regenerate 6 RUBP for 6 cell cycles to continue. Seems complicated, but yes, this is how it goes. Apart from that, this Rubisco enzyme uses the pH of 8. So the pH 8 is an alkaline pH in the stroma. It works well in that pH only. How is this pH maintained? The pH is maintained by non-cyclic photophosphorylation, because there's a high H plus concentration in the thylakoid space and a low H plus concentration in the stroma. The H plus that diffuses through chemiosmosis is used to produce the NADP in the stroma. There are no free H plus in the stroma, otherwise the pH of 8 would not have been maintained. The pH would have reduced. So this is what I want to discuss in this video. I hope this was valuable for you. Thank you so much.